Hello, my name is Peter Caravan, and I'm an Associate Professor of Radiology at Harvard Medical School and at the Massachusetts General Hospital. Today I'm going to talk to you about thrombus and detection of thrombus using imaging. So what is thrombus? Thrombus is a blood clot that forms in the arteries or veins of the body. And this is generally a very dangerous phenomenon. It can cause stroke, it can cause heart attack if it's formed in the coronary arteries. It can cause death and shortness of breath if it's a blood clot that forms in the lungs, or it can be very painful if it forms in the deep veins of the legs. Thrombosis affects millions of people in the world, and it costs the healthcare system billions of dollars worldwide. We need better imaging tools to diagnose thrombosis and to monitor its disease progression and its resolution. So how do we do that? The way that we went about doing this was to look at the biochemistry of blood clotting and the coagulation cascade. And we identified fibrin as a protein that we really wanted to target as a way to visualize blood clots. So fibrin is a protein that's only present in blood clots and in wound healing, but it's not found in circulating blood. And because of that, imaging fibrin should provide high sensitivity for disease, but a very low background. Now the challenge in imaging fibrin is its biology. Where does it come from? So fibrin comes from a protein called fibrinogen. Fibrinogen is circulating in your blood at a very high concentration. It's sort of like a sentry that stands on guard for blood clotting anytime that there's an insult to the body. In the presence of damage to a blood vessel, an enzyme called thrombin will activate fibrinogen, will cleave some small peptides from it called fibrinopeptides, and then this will form a fibrin monomer. The fibrin monomer then undergoes a polymerization reaction. So the fibrin monomers undergo this end-to-end formation of a chain to form a fibril. And then there's another enzyme that gets involved called factor 13, and factor 13 serves to cross-link the fibrin strands. And this provides a very stable matrix, a very stable net that causes blood to clot. Now the challenge for us was really how are we going to be able to find a molecule that binds to fibrin but does not bind to fibrinogen, which is a high background signal. So we turned to a technique called phage display. Phage display is a way of looking at a number of different peptide sequences that are expressed on the surface of a bacteriovirus, a bacteriophage. So the way that we did this was we took a library of these bacterioviruses, and all of these viruses had a different peptide associated with it. And then we took some fibrinogen and we incubated the fibrinogen along with the phage library. We took all of the phage that bind to fibrinogen and we removed them from the library. And in this way we got rid of things that were going to cause background signal. Then we took our remaining phage library and we incubated that with fibrin. We washed away the phage that didn't bind to fibrin, and then we dissociated the phage that were bound to fibrin. So now we took this library of about 1 billion different phage, and we winnowed it down to a very small number. Now since phage are virus and they're living, we can incubate them with bacteria, grow them up again in culture, and run through the process again. And by doing this over and over, we were able to engineer peptides that had high specificity for fibrin and low affinity for fibrinogen. We then characterized what these peptides were. So now that we have a peptide that targets fibrin, we need to make it visible to an imaging technique. And the imaging technique that we're going to use is magnetic resonance imaging. This involves a large magnetic field. And when you put a subject into a large magnetic field and apply a radio frequency at an FM radio type frequency, we're able to detect hydrogen atoms in mobile molecules. And these are mainly water molecules, but also in fats. Our body is largely comprised of water, and this is what we can detect. So the contrast that we see in an MR image arises from the fact that there's different concentrations of water in different tissues in your body. But water also has different magnetic properties in different tissues, and we can exploit that to create image contrast. Another way that we can create image contrast is to use a metal ion. In this case, we use a metal called gadolinium. The gadolinium 3+ ion has 7 unpaired electrons, and this creates a very strong local magnetic field as it tumbles in solution. But this is a toxic metal ion, so we need to sequester it. We need to bind it very tightly with a chelator to prevent the release of the toxic metal ion. But we can do that, and we can do it safely. And these types of molecules are injected daily into patients in hospital to provide MRI contrast. They're rapidly eliminated from the body after we get our imaging exam. Here's an example. This is a woman who has breast cancer, and what we'd like to understand is whether the cancer is spread and metastasized to the brain. The image that you see here is an MRI before we inject our contrast agent, and the image here is now an image after we inject our contrast agent, and you can clearly see a region of signal enhancement that appears, and this is due to cancer that's spread to the brain. So the next step was really to take these two things and put them together. We've got a peptide, and now we need to link to it these gadolinium chelates to provide MRI signal. And this is sort of an iterative process where we want to link the chelate in a way in which we don't impact the binding to fibrin and don't impact its metabolic properties. We did this, and then we had a molecule now that was sensitive for detection of thrombus by MRI. But we could change the label. Instead of using gadolinium, we could use a radioactive isotope and detect it using a technique called positron emission tomography. Or we could change the label to a fluorescent probe, and we could image this optically with fluorescence microscopy. As you can see here, this nice image of a fibrin mesh. The molecule that we ultimately created was called EP2104R, and it was very specific for fibrin over fibrinogen. It doesn't bind to the other proteins in blood plasma, and in this way we have a high target to background ratio. It has something called high relaxivity. It's very bright in an MRI image, and that's because it's got 4 of these gadolinium chelates in the molecule. It's completely eliminated from the body, which is very important because we want this metal ion to be eliminated from the body and not cause any toxicity. It doesn't impact blood clotting, and it doesn't impact the dissolution of blood clots. It's just an observer of blood clots. And it was well tolerated in patients and in animals. This is an example just showing you how it works and how we test these molecules before we take them forward into human trials. In this case, our goal was to mimic a model of stroke. And so what we did was we generated a blood clot, and we delivered the blood clot via a catheter to the internal carotid artery of a rat. And what this does is it stops blood flow to the brain, and you can see this here on this image. So on the left side of this image, you see a number of blood vessels that are enhanced, and on the right side, there's an asymmetry. There's not much enhancement on the right side. And this is because the blood flow is blocked to that side of the brain, and this is what causes a stroke. Now, when we gave our molecular probe that targets fibrin, here's the images that we saw. The arrow points to a very bright focal area in a blood vessel, and that's the presence of the thrombus. You can see it here on the left on this orthogonal image, and the image on the right is in plane, and you can see that the thrombus extends through the internal carotid artery and starts to branch into the middle cerebral artery. And we don't see any enhancement on the other side, in the vessel on the other side, indicating that this is specific for the thrombus. And going from there, we went further into patients. And this is an example of a man with a history of deep vein thrombosis and now has clinical signs of deep vein thrombosis. So this is signs of a blood clot in his thigh. And the image on the left is an MRI of this man's legs before we inject the probe. And the image on the right is an image after injection of the probe. And you can clearly see where I've marked it with arrows, region of very high signal intensity. And this is a very long blood clot that extends up the thigh. And in this last example, this is a woman with a history of heart disease who has the presence of a blood clot in the right atrium of her heart. And that's shown here with a red arrow pointing to this bright spot. The yellow arrows in the image that demarcate what looks like a large tube, this is actually the aorta, and there are blood clots that line the wall of the aorta along some atherosclerosis in this patient. What I conveyed to you today was how we in the lab go about addressing unmet needs in medicine. In this case, we wanted to identify a molecule that allowed us to visualize blood clots anywhere in the human body. And in order to do this, we went back to biochemistry. We looked at the biochemical constituents of blood clots and identified a protein, in this case fibrin, that was only found in blood clots and not in the circulating blood. Using a combination of biochemistry, molecular biology, chemistry, and imaging, we identified a molecule that could safely detect blood clots in humans, and today is being evaluated in human clinical trials.